Hello, I'm Jim Cornell, and this is Base to Base, the weekly biotech podcast from Air Coastal Media. Yes, welcome again to the Base to Base biotech podcast, and this is number 39, going out on December the 12th, and it's another 3-interview podcast, a bit like the 3 advent calendars in our house. Speaking of which, it's Gingerbread House Day today. I suspect with food prices rising, it may be more expensive than one made from bricks very soon. Last week we had conversations with Shanna Bosma, International Relations Officer at Health Holland, Mark Bagnall, CEO of Phenomics, and PDS Biotechnology CEO Frank Bedu-Addo. This week we're talking about an artificial intelligence event in Sweden with Jakob Andersson, as well as about genetic medicine for eye conditions with Coave Therapeutics CEO Rodolphe Clerval, and there's a conversation with MOBILion Systems CEO Melissa Sherman. If you do have news or suggestions for who should be on the podcast, or indeed any other comments or suggestions, please do get in touch at jim@deeptechdigest.com, which is also the address you can use if you're interested in advertising with us or sponsoring a podcast episode. First, let's take a look at the news you may have missed over at deeptechdigest.com. And it was another busy week. A new RNA class has been discovered, lower doses of immunotherapy for skin cancer give better results, and OncoInvent published its Phase 1 ovarian cancer results. A 7,920-acre next-gen industry facility called IBTech has launched in Malaysia. A new CRO has been launched called Avetra, and Espirare received a Gates Foundation grant to advance a preeclampsia novel therapy. There have been positive results from an ALS study, as well as from a male and female infertility monoclonal antibody trial. Researchers have seen potent and selective anti-tumor activity in an acute myeloid leukemia study. Chemlex raised $45 million and launched a lab for drug discovery in Singapore, and a new CRS program is going to feature Poolbeg Pharma's trial. And while the endpoints were not met, new data provide insights to Alzheimer's pathobiology. You can read all of these and plenty more at deeptechdigest.com. Okay, let's get to the interviews, as that's what it's all about. And this week, first up, is a chat about improvements in genetic medicine being applied to treatments for eye conditions. The company spearheading this is Coave Therapeutics, and we had the opportunity to find out about how the company is making a difference with the CEO, Rodolphe Clerval. I wonder if first then maybe you could tell me a little bit of the history of the company.
Sure. So the company has quite a long history, actually. The company started in 2015 under a different name. The company was named at that time Horama, and it was already a gene therapy company, but focusing on one single ultra-rare disease, which is one form of retinitis pigmentosa. And I've joined the company where their first product was just starting the clinical development, and we completed the clinical development of this product, and we licensed out this product to a partner, and we have refocused the company on still ophthalmology, but trying to address more prevalent disease with different technology of delivery. The first product was based on I would say the first generations of gene therapy product, and now we are working on the third generation of gene therapy product. So that the company was created from University of Nantes in the west of France. And when I joined the company in 21, so basically 6 years after the inception of the company, we restarted the company in some extent with a different name, a new investors on board, and but still in the field of gene therapy and ophthalmology.
But why did you change the name?
Because of the technology, we really wanted to anchor the company position around this technology, so the classical technology used in gene therapy to deliver the therapeutic gene is called AAV, for associated adenovirus vectors, which was a technology used in the first generation product. But we have modified this technology to put what we call ligands, so targeting moiety at the surface of these AAV vectors, which are a virus. And the way we attach those moiety to the surface is by conjugation. So we call this AAV conjugated AAV. That's why the company name is coAAV, COAVE. Just it's a nickname of conjugated AAV vectors. That's the whole story. And now everything we are developing is based on this conjugated AAV technology as a primary delivery component of the therapeutic product.
Could you give me a bit of background on retinal vascular diseases and what they are and the differences between the different ones and how many people are suffering from those conditions?
So it's a prevalent condition. There's what we call them the age-mediated retinal degeneration, AMD, and there's wet AMD, dry AMD, basically. The wet AMD, which is one of the most aggressive and damaging form, is basically wet because in the back of the eye, the retina, where basically all the pictures we can see are transformed to an electrical signal to the brain, this kind of screen. Here, there's another growth of vascular cells and tissue. So there's a lot of vessels that are growing, and that leads to actually leaking of fluid. So you have an accumulation of fluid, what we call edema, and those accumulation damage the cells of this retina, and those cells cannot be renewed. We call those cells photoreceptors. And basically, when you lose them forever. So what happened to these patients is they start to have a blurry visions, and then they start to have black hole in the center of their visions that increase during the disease. And this is because the growth of the vessel. And the mechanism is very well known. Those vessel growth is activated by one factor called VEGF. And the way to prevent this vessel to growth is to block these factors. So 15 years ago, drugs have been discovered and used for the last 15 years, which are antibody binding to these factors, VEGF, which are proven to be very efficient, actually. So the way those antibodies are used is you inject them right in the eye using a syringe. It's called the intravitreal administration. So you go to the physicians. It's a very rapid and pretty not painful procedure. But you have to do this injection every month or every 2 months for the rest of your life. And this is one of the major burdens because those products work. They are so complicated for patients to be used that patients tends to not get these injections as frequent as they should, or some of those patients, actually almost a majority of them, will drop out the treatment after a few years. So there's current standard of care in this disease. We know how to treat it, but we think we can do better with gene therapy.
Are there any other current treatments? I've interviewed companies that are doing work on wet AMD and dry AMD. Is there a lot of options?
So for wet AMD, there's quite some options. There's actually 4 treatments available on the market, all working the same. I mean, there's no difference in terms of efficacy. The main differences between those treatments has been the durability. So how frequent you have to use the treatment. 15 years ago, the first treatment was every month. Now, the last one, the first treatment is Lucentis from Novartis. The last one, Vabysmo, which is an antibody basically targeting the same mechanism, has demonstrated that you can increase this interval by 2 months, maybe 3. And the durability improvement is what everyone is looking for. But 3 months is still not enough, because when you look at the real world data, so now, institutions and university, they can capture all data from patients in the real life. What we see is basically after 3 years, more than half of the patients are basically dropping out the treatment, and the remaining half are extending their interval so long that it impacts their vision. So the bottom line in this large real life or real world data is the patients, every gain they obtain in the first few years because of the treatment is vanished after 3 to 5 years. So they are back to zero. They are back to losing their vision. So on the long term, what happens is those current biologics and treatment available are not providing what the patients need. It's basically preventing their loss of sight and visions, and this is where many companies are looking for alternatives to really address this issue of durability.
So what has your approach been to tackling this?
So our approach is gene therapy, and gene therapy to deliver the gene of fragment of antibody that are commercially used to target this what we call angiogenesis, so the anti-VEGF and other factor that affect the growth of vessel. So the way it's, instead of injecting the antibody, already the protein, we inject the vector we are using, the conjugated AAV vectors that have a unique way to go straight to the retina, to the back of the eye, and there they deliver a copy of two genes. One is an anti-VEGF, the other one is an anti-Ang2. Both are working on preventing the growth of vessels. Those genes will be expressed by the cells that lay on the back of the retina, the RPE or photoreceptors, and they will produce this fragment of antibody, and what we call a biofactory. They will produce the antibody constantly in the back of the eye to capture and block the factors that lead to growth of the vessel. And by doing so, there's many interests. The first one is you need one injection at the beginning, and then you have a constant expression. So what we expect is over a year, more than 70% of the patients that will receive our treatment will not need additional injection. So one injection and constant expression. This is a perfect durability optimizations that everyone is looking for. The second interest is because you have this constant expression of these antibodies to block those factors, you don't have these fluctuations that happen with the current biologics, where you inject, you have a certain level of antibody and they disappear. So what happens is the growth of vessel restart again. So you inject again and you stop it, and those fluctuation leads to more damages in the retina. By having a constant expression of the antibody in the back of the eye, we know it will lead for an improvement of control of the growth of the vessel. And the last very important component is the safety. Why? Because this approach of using gene therapy to deliver the gene of antibodies to do this biofactory, we are not the first one to think about that. There's other companies currently in clinic having different versions using the first generations of AAV vector to deliver. What those companies have shown is this approach using classical AAV and the route of administration they are using is leading to substantial inflammations that require the patient to be put under anti-inflammatory treatments, what we call steroids, for quite a long time, 5 months. This is a burden for the patient. So basically, you propose those patients a gene therapy to replace multiple injections, but those patients will have to take multiple treatment for five months to maintain the inflammation caused by the gene therapy. As opposed to that, our gene therapy has proven to be much less inflammatory for two reasons. One is because the route of administration you are using, it's called the suprachoroidal route. It's basically a way to avoid having a direct inside the eye injections, but more around the eye injection. And the second aspect is our conjugated vectors, our technology, by the presence of these ligands at the surface, create a shield against the antibodies that come from the body and trigger the inflammation. So in long story short, it's a gene therapy, but it's a second or third generation gene therapy that deliver antibodies or will lead to the constant delivery of antibody in the back in the eye with a very good safety profile.
Excellent. You recently presented data at the EGSCT Congress. Could you tell me a bit about that?
Sure, there are very important data because there are data showing how efficient is our conjugated AAV to reach the key cells in the back of the eye. This data we presented is actually the third non-human primate studies that we have conducted using this technology. And what we confirm in this study, what we have seen in the previous one, is our conjugated AAV with specific ligands using this route of administration, the suprachoroidal, has proven to be 25-fold better than the classical AAV used by competitor to reach the back of the eye. And moreover, it's 25-fold better, but we see an amazing spreading. The back of the eye is like a globe, so you have to be sure that you go all around this globe to be sure that you target all the cells. We have a unique spreading in all those cells, and we can penetrate and transduce. So basically, the DNA is delivered not only on the first layer, but also on the upper layer of the retina. So what we call the RPE, the epithelial cells of the back of the eye, plus on the top of those cells, the photoreceptors, which are the key cells that we want to protect. So we have demonstrated unique delivery and efficacy of delivery features of these vectors. Plus, in this primate, we have seen that there were no safety issue with these vectors.
Could you tell me a little bit about your clinical trial pathway and where you're at with that right now?
Sure. We are aiming to have an IND by the end of 27. So we are still a little bit far from this, but what is important is in the next few months, we'll be treating the first primate with the final product, which is this capsid that I described before, these vectors, plus the key DNA piece we put inside, what we call the cassette. And this product will be tested in primate to demonstrate how much of antibody we can produce in the back of the eye. And in this disease, wet AMD, the level of antibody produced in primate and the efficacy is a model that is known to be extremely well predictive of what happened in humans. So that we're going to have actually two NHP study in 26. One is pharmacokinetic. We look at the level of anti-VEGF antibody. And the second one is a dose response, where we look at which dose, which efficacy we see. Those will be two critical primate on our path to IND, two critical study for our to IND, because they will validate that the product worked again on a well-known model in a large animal model. Then in end of 26 and 27, we will engage the IND enabling study. So basically the GLP tox and all the work that should be done to prepare the package for filing an IND in US and a CTA in Europe for having the first patients being treated early 2028.
How important is it to be able to treat early? Are you able to completely cure somebody who's far along with wet AMD, or does it rely on them only being diagnosed early?
It's relying on them being diagnosed early, because our product and mechanism will not actually enable the patient to recover their vision loss. This is not a reparation that we are making. It's really preventing the disease to really expand. But, this is a disease where I didn't mention the numbers, but we have approximately 1.5 million patients in US and 2 million patients in Europe having this disease currently at the time we speak. So it's a very frequent. So the ophthalmologist knows how to detect this disease. Basically, the patients, they start to see small dots and blurry visions. They go to the ophthalmology office, and very rapidly now this disease could be extremely well identified. Then now the process is those patients are treated directly with the anti-VEGF treatment available. It's done directly by the ophthalmology. It's an in-office procedure. You don't need to go to the hospital. And so the field is aware about the importance of treating patients early. But what the field is aware too is those patients, after 1 year, you really have to keep them under treatment. If they start having long interval because they don't want to get back, because it was last month and because they need often someone to bring them to the office of the physicians. That's where the issue is. So what we do expect is actually very rapidly when the patients will respond to the first biologic treatment, the physicians will talk with the patient and say, look, for the next years, instead of continuing the frequent injections, we're going to switch you to one injections. And it should be done. We're going to have them in surveillance. Maybe we're going to do one injections every 2 years to just keep you at the level. But we have this opportunity. And this is exactly where we want to be in this position, to be the backbone anti-VEGF treatment, but, using gene therapy or technology that will, avoid patients returning 6 to 8 times a year at the physician office.
Is there anything else in your pipeline that you're working on?
So we are working on actually in ophthalmology, other products using the same delivery technology, but for one form of dry AMD. We discussed briefly dry AMD, but dry AMD, you don't have real treatment currently. There's some form of dry AMD that are called geographic atrophy, where you have actually visible atrophy of the retina. So we are working on discovery level on new programs. And beside that, we are working still on our technology of delivery, but for other tissue. We have actually vectors that enable to target the brain, which is a tissue very close to the eye in terms of how does it work, to target the brain from actually a direct injections in the bloodstream, an intravenous administrations, IV, and vectors go to the brain by what we call crossing the blood-brain barrier, BBB. And again, we're using the same technology with those vectors decorated with ligands at the surface. Those ligands will target specific receptors on the BBB and crossing the brain. And with those vectors, we can address multiple diseases, from rare monogenic disease until prevalent disease like Alzheimer and Parkinson. So this is still early, but we think we have the right technology to address this kind of indication.
All right. Those are the only questions that I had. Is there anything that you wanted to mention that we didn't cover?
Those days, it's interesting because we see over the last 6 months, really the gene therapy, especially in ophthalmology, coming back to the front of many pharma and investors. We have seen many companies raising round. We have seen company being acquired, especially in ophthalmology, because this is the most adapted tissue for gene therapy. You still inject a low dose. You don't have systemic toxicity issue because the vectors you inject remain around the eye. And this is the only indications where you can treat rare disease as well as prevalent disease like WND. So we have seen the field really, it's a renewal of the field, because the last years in gene therapy, we suffered like many other modalities from, investor skepticism. I think we are beyond that now, and it's a very good news. And we are pleased to be part of this, and especially pleased for the patients to be able to bring new product for them because they need them.
Not necessarily an exclusive problem for the aging population, but it is interesting to hear about new treatments that remove a little bit of the burden for patients. As I've always believed, it's no use developing fantastic treatments if no one is going to want to participate. Next, we're headed back to Sweden. While I was at NLS Days, I got the chance to learn about the GAIA Conference, which is about AI and how the AI industry is helping life science companies and some of the challenges being met. I headed over to the Tech Week event early in the morning, thanks to Business Region Gothenburg, and while there, I had a quick chat with Jakob Andersson, Vice President of Engineering at Swedish company Anaphora and founder and chairman of the Gothenburg Artificial Intelligence Alliance, or GAIA, which hosts an annual conference in the city on AI. I guess first, if you could tell me a little bit about yourself and what you do.
So my name is Jakob Andersson. I'm the founder and chair of GAIA, the Gothenburg Artificial Intelligence Alliance, which is a non-profit working with building a community within AI in Gothenburg region. I'm also the tech and product lead at a small startup called Anaphora here in Gothenburg.
Could you tell me a little bit about both of those? Maybe start with the company, and then we can broaden it out a little bit.
Sure. So at Anaphora, we work with brand governance. We work with big iconic brands that has a distribution model where a lot of people need to touch the brand and communicate with customers. And we make that easy to stay on brand. You when you create an email or an event, you can only be on brand. So it's super easy, and for the company, you know all your employees adhere to your brand, and the experience for the customers is great all the way. So that's what we build. We're looking to scale that today, both internationally and to other industries as well. At GAIA, we started as a community back in 2014. A small meetup group around machine learning and data science here in Gothenburg. When AI started really to take off and like the big hype, we decided to create a non-profit organization and a conference around this. So that's back in 2018. We had our first conference that year, had a big success and must have grown since. Despite the COVID pandemic and everything, we've managed to continue growing that, and we're next year, on April 20th, we're looking to have 1,200 plus attendees here in Gothenburg across 3 different tracks, all covering machine learning, data science, data engineering, but also on the adjacent topics like ethics and design and business, legal, and all the things you need to understand and be aware of if you're building an AI company or just working with AI products in general. We want to be for the practitioners in the industry.
And what are some of the challenges that companies face with implementing AI? I guess that there's a range from knowing nothing to a lot.
Yeah, definitely. And I think the speed of AI and the change is incredible. And there is a clash in that in a sense where you have the traditional way of working and planning for a long time, and then you have the super dramatic changes happening over a month where you kind of need to change how you think to actually ride this wave and not be left behind, regardless if you actually had the right ideas or not, because the speed is so different. So that's definitely one thing. Then finding the right competences is another one. There's few who have done this in the past for natural reasons, but there are a lot of people talking about it, and distinguishing who is which is definitely a challenge too. And all the changes to the business models and everything, it's many things at the same time. It's kind of a perfect storm, if you will.
What's your role in helping companies not only to start with AI but to help them through? Because I think sometimes companies think that they need AI, they get somebody in to set something up, and then they're on their own.
Yeah, definitely. And we kind of take two perspectives on this. For the companies and leadership, we want to educate and go beyond the hype and like understand what it takes to build an AI system, both that it's not necessarily that much every time, but also that you can't just believe everything is possible. It's not magic. You need to have a bit more nuance in what you believe is possible and how you educate your workforce and that. So that's one part. The other is on the engineering side or development side, that creating more balanced engineers. I think that's really important for especially startups, but also bigger companies, that the people with expertise in the subject, in the machine learning, in the data science, that they also understand the context around, because they will need to lead their companies. They need to understand that they can't just go with the technology that seems fun because it may have ethics problems, or it may have legal problems, or the users won't understand that way, or all those things. So creating those more well-rounded engineers and developers is also a really key part of the puzzle.
And are there some people have unrealistic expectations of AI?
Definitely, but also the other way around, that people have too small expectations. And I like to quote William Gibson on this, that the future is already here, but it's not distributed. I really think that there's so many things that's possible, and especially in the near future, but also a lot of things that people believe is possible, and especially in their organizations that's not possible, because it's also the transformation journey. Same with IT and everything in the past. As a startup, you can take big leaps because you have nothing to change. But in the big company, you need to do that too. And at the speed of change in AI, that's really tough.
One of the things that in the life sciences especially that is looming as a potential challenge is going to be commoditization of data, where the biggest companies have access to it and smaller companies don't. Mm-hmm. And it either creates a two-tier system, or it creates a system whereby the only way you can get access to data is to pay for it. Are there challenges like that you see in other areas other than life sciences, and other challenges maybe that we haven't encountered yet?
Yeah, that's definitely a challenge. I read an article recently about the big tech giant Google and Microsoft and Apple, and those being the ones guarding the data from many parts of AI. And like you say, it's about having access to the data, otherwise you can't do anything unique. What AI is doing is that it's changing dynamics in what's costing in developing a product or a service, that previously maybe software development was the big cost, or maybe the marketing was the big cost. But when AI accelerate those things, other things become the critical part, the access to the data, so you can actually do the service or the product, regardless if you can do it in the future automatically with some incredible AI developer or not. If that's fast and everyone can do it, then it's the one who actually have the data as the foundation for the system that can do it. And that's really a challenge in many industries. And for a company, you need to secure that you have something that's unique in that sense, that you can defend compared to your competition. Because if you just create some service with the latest tools, and anyone could replicate that. So you need something that's unique. But that's also, like you say, kind of a gatekeeper for those that already have the data. Definitely a challenge.
And what about security? Is that another major challenge?
Yes, for sure. And both for private individuals and for companies, and the acceleration of exploitations and creation of viruses and security threat is incredible. And I mean, in the past, maybe you took a couple of months from new flaw being discovered in a software until it was exploited. Now it's a week or maybe less, because it's automated by AI. And the same with phishing, that in the past maybe you had someone calling your grandmother or trying to say that they are from the bank or similar. But today, with just tiny pieces of information from Facebook or public platforms, you could create a video call actually looking like your grandmother, or with the correct voice and information about the latest holiday, so you can perfectly imitate a person or like a CEO at the company or something, and that's incredibly challenging. Since security is always about the weakest link, you need to think, how can we as a company or private individuals or a government educate everyone to be vigilant to this, and it's an incredible challenge.
And how important are events like the one today and other events that look at AI?
Yeah, I think they are crucial because knowledge is the foundation. If we don't understand what's happening, and we in the tech industry, we need to be the ones spreading the knowledge. We need to educate ourselves and network and build communities that can help each other. It's a challenge for everyone, and it's a challenge for society, so we need to educate ourselves. So events are really crucial for that.
And are they also important because obviously you know what you're doing, and a lot of attendees to events are kind of at the other end of the scale. Is it important to kind of understand what the questions are?
Yeah, for sure. And at GAIA, we have broadened our audience quite a lot recent years. When, say, after ChatGPT, that's kind of the defining moment in the public, where everyone's starting using this system and asking questions, and we need to answer those too, and create events that not only target PhDs in machine learning or like the people working here in 10, 15 years, but we need to help everyone and educate, and that could be teachers in schools, it could be users of AI systems in finance or whatever. Everyone needs to be aware of what's happening, both the potential but also the risks.
It's amazing how quickly AI has infiltrated our lives, both personal and business, and it's interesting to learn of some of those challenges that are still being faced, as well as how more companies can incorporate AI into their own operations appropriately. Next, we're talking about separation science and multiomics, and another way life sciences companies can get help in their operations, with Melissa Sherman, CEO of US company MOBILion Systems. Maybe you could start off by telling me a bit about the company.
Yeah, so it's a pleasure to be here. Thank you for the opportunity. So MOBILion is the company that I lead. It is a separation science analytical instrumentation company that is commercializing a cutting-edge science that was licensed from a U.S. federal lab to change, advance, disrupt the analytical instrumentation market. So specifically, or I say, if you remember one thing about what we do, we say in our tagline, we reveal what others leave unseen. And what that means is in our space is liquid chromatography, mass spectrometry, analytical characterization workflows. So LC mass spec is kind of the gold standard incumbent analytical characterization workflow for a variety of molecule classes. And we are addressing, I'll say, the deficiencies of liquid chromatography from a separations perspective with our technology to give better analytical results faster, with greater confidence, greater specificity, greater reproducibility. So we basically see things that LC mass spec cannot, and we see those things faster and more accurately.
All right. I guess there's still a bit of confusion around the different omics or omics terms. Could you maybe clarify some of those? Because I think there's an omics for everything now.
Yeah. Well, I think understandably it can be very confusing, and I think it's really understanding the holistic scope of the biological processes, right? So genomics, DNA makes RNA, which makes proteins control cellular function. So it's almost like I think about it as a jigsaw puzzle of the different omics, right? Proteomics, lipidomics, metabolomics, glycomics, and all of these are potential measures of the function of a cell. And so if genomics or DNA is the blueprint, if you think about, let's say, I hear people use the analogy of making a cake. if you make a cake and you follow a recipe, that doesn't always mean that the cake turns out perfectly. And there can still be things wrong with that cake once you make it and you bake it if something goes wrong. So genomics is the blueprint or the recipe. Measuring all the other omics gives you an indication of how successful or how right or wrong those biological processes are. And so, the other analogy is thinking about a jigsaw puzzle. And if each piece of a biological process is represented by a piece of that jigsaw puzzle, you don't get a complete picture of that biological process unless you have all the different pieces. And so each of the omics is really a different piece of that jigsaw puzzle that you put together to create that complete picture of human biology. And so, when you think about, well, what does that mean and what impact it has from a disease diagnosis and treatment perspective, the more accurately and deeply you characterize the patient's biology, and the more accurately and deeply you characterize the drug chemistry that's being developed to target that patient biology, the better disease diagnosis and treatment system you have. And so when it comes to that target biology of the human and understanding cellular function and mechanism of action, you need to understand that in order to develop the right drug target that targets that cellular function and that mechanism of action. And so it's really two sides of the coin, right? It's almost like a Venn diagram coming together. You have to completely characterize the biology that you're targeting or try to affect. And then you also have to more completely characterize the drug chemistry so that it's right and appropriate to target that biology. And all the different omics impact that characterization of either the human biology or the drug chemistry. And so, when it comes to earlier disease detection, more personalized disease detection, better understanding of disease mechanisms, which ultimately leads to improved drug development of safer, more efficacious drugs, we need to be able to more accurately and more quickly characterize all those pieces of the jigsaw puzzle. And that's where we come in. So in advancements in analytical instrumentation, there's always an incumbent, right? There's always, this is the way that people used to do things. But understanding and appreciating that there's always room for improvement. So, don't get me wrong, LC mass spec is the gold standard of characterization downstream from genomics. So proteins, lipids, metabolites, glycans. Liquid chromatography mass spectrometry workflows are the gold standard. That doesn't mean that there isn't room for improvement. So what is, some of the issues with the incumbent liquid chromatography and mass spectrometry workflows are that they can be slower, they're very complicated, can be finicky, and they miss things. They just miss things. And so advancing that characterization, which is what we do with our platform, we go faster, it's less complicated, it's more automated, it's more accurate, more reproducible. So you see things faster, and we see things that other instruments fail to detect to give you greater confidence when you're characterizing either human biology or drug chemistry.
I think everybody, well, most people have been in a situation where they've been at work and somebody has said, we've always done it that way.
Right.
And you don't really make any progress by sticking with that, because until you start to think, well, how else could we, or how can we take it further? How can we do it differently? I don't think you make any progress until you think outside that box.
Right. It's an exciting opportunity to be at the forefront of breaking through barriers and catapulting the quality of analytical characterization. It's also difficult because it's a conservative-ish industry, right? And we have always done it that way. And oh, by the way, there are FDA submissions that include data for how it was done in the past. So the last thing you want to do is challenge what you've done in the past that you've got an FDA submission relying on, right? So it's a challenge, but it's a hurdle that needs to be overcome, which is what just requires patience, right? So in the discovery, and as people and our pharmaceutical customers use our product to make discoveries, and they talk about, oh my gosh, we didn't even know this was possible. And wow, we knew from the purity of the theoretical chemist that there were two forms of this drug, and one of them is efficacious and one of them is not. And we did not have a way to separate those two compounds from one another before. It's incredibly rewarding to now show them you were using this analytical technique and you actually have blind spots. Some are known and some are unknown in terms of these blind spots. And uncovering or addressing those blind spots will only lead to the development of more efficacious and safer drugs. It's a challenge because it takes a long time to convert people, and it takes a long time to get people to see you thought that was the characterization profile of your, mechanism of action study or your drug study, and actually there was error in it. And so it opens up the opportunity. It takes 12 years and $2 billion to bring a drug to market. Well, there's no wonder why, right? If there's inaccuracies along the way, it's failed clinical trials. It's not fully understanding the mechanism and the drug chemistry that leads to these failures and leads to these inefficiencies. So if you can address that, even in just the littlest bit, you can save hundreds of millions and billions of dollars in time in the drug development process holistically.
For those that don't know, myself included, could you tell me a little bit about the technologies and the definitions of high-resolution ion mobility separations, and SLIM, and all of those good things?
Yeah, so SLIM is the name of the core technology that we licensed from an inventor, Dr. Richard Smith. So Dick Smith is a researcher in the analytical instrumentation world, mass spec world, that's at a U.S. federal lab. His research group is at a U.S. federal lab. We, MOBILion, licensed Dick Smith's technology, which is the SLIM technology, and the patent portfolio associated with it. And it's just a category of high resolution ion mobility. And so, it's a mouthful. And without really understanding the science, the easiest way to understand what we do is, from a separation science perspective, the incumbent is liquid chromatography. And if you just envision liquid chromatography as plumbing, liquid, pumps, solvents, tubing, and you put your sample into liquid chromatography and it gets separated through columns, through chemical interactions with the column in liquid phase kinetics. We say with our technology, and I use this little card, so this is a printed circuit board. It says SLIM on it. It's a printed circuit board. So this is the form factor of our separations technology. We say we digitize separations. So when I think of it, I kind of think of, remember old school cameras that had rolls of film in, and you had to send your film in to use wet chemistry to process the film, and then you got your pictures, I don't know, a couple weeks later, and some of them would be good and some of them would be bad, and there was nothing you could do about it. And high quality photography was reserved for trained photographers. Well, then you digitize photography and you put it on your phone, and now everybody's a photographer. So you've democratized photography and you get real-time answers. You see the picture real time on the screen and you can retake it, or you can, make changes to it. So digitizing separations and taking it from that complex liquid plumbing, solvents, and tubing kind of situation and using electronics and putting it on printed circuit boards just means that you can democratize separations, make a very complex analytical technique amenable to less sophisticated operators, and make it more automated, and make it more efficient, and make it faster. So high resolution ion mobility just says on a printed circuit board, we move ions using electronics to achieve separations that liquid chromatography can't achieve. So we're in the gas phase, which means we're in gas phase kinetics. So we're 1,000 times faster than liquid phase kinetics. And then because of the way we separate based on inherent physical properties of molecules instead of chemical properties, we achieve greater specificity, greater accuracy, and greater reproducibility. So with electronics, analyte agnostic, we separate on printed circuit boards much faster, much more accurately, and much more efficiently. And the reason it's called high resolution ion mobility is just that we ionize the analytes and we use electronics to move the analytes through the printed circuit board to achieve separations. in a high resolution or high resolving power configuration.
So you mentioned the photography analogy. Does that mean that Slim is going to be on everybody's iPhone within a year?
Well, maybe not the iPhone, but it's going to be on everybody's mass spec. So we integrate with a mass spec detector, right? So a mass spectrometer is the detector. So much like it, right? Not only do we, in terms of advancing the industry, putting separations techniques in more users' hands because it's simpler and more automated is certainly our goal, much like putting photography in everybody's hands, right? And just removing the complexity and increasing the throughput and increasing the accuracy, we attach to mass spectrometers as detectors. And certainly because we're working with a printed circuit board form factor, we can customize it and tune it for all flavors of mass spectrometry and pretty much all applications where mass spectrometry is used. So we're talking about life sciences and pharmaceutical industry, but it's also used in food characterization. It's also used in environmental testing, and there are a lot of different types of mass spectrometers as detectors. We do hope to integrate with all of them from all vendors to really advance the LC mass spec industry as a whole.
Could you explain your platforms and what they do and how that fits in with a lab and ultimately helps your clients?
Yeah, so we have two products that we've publicly announced in our roadmap. The beauty of a technology like this is that we can change the printed circuit board pattern and just with electronics telling the analytes to do different things. The sky is the limit in terms of the different products that we can develop for different applications, fit for purpose, depending on whether you want a clinical analyzer that's used in a hospital test lab, you want an instrument that's used by a pharmaceutical company in discovery, or an instrument that's used in a QA/QC lab further downstream in the value chain. All we do is change the electronics and the printed circuit board patterns, and we have instruments that can do all things for all people. And so, the sky's the limit in terms of us for the product roadmap that we have in our development pipeline. Our first product that we launched commercially was integrated with a high-res mass spec to do more targeted analysis for food characterization, environmental characterization, and drug discovery. We have a second platform that's in development that we've talked about that's not yet commercial for the proteomics space. So when you're doing a proteomics discovery, you want to see tens of thousands of proteins and all the associated PTMs. That's a different platform and a different requirement than if I want to look at 100 metabolites in a metabolite screening workflow. And so the latter, 100 metabolites, 3 different drug conformations, when it's a more targeted workflow, we have an instrument for that. And that's MOBI, our MOBI platform. And then we have a product in development that's our Billy platform that is for proteomics discovery, really multi-omics discovery. And that's when you want to look at tens of thousands of things and you want to see all the things. What connects the two platforms, regardless of end use and regardless of application, is we see more things faster and more accurately in both cases. And so we're really excited about our proteomics platform, and we're working with a number of vendors to productize and commercialize that platform because that really advances personalized medicine and really advances science from an early disease detection and improved drug development perspective. What
kind of challenges are your clients facing and how are they using your technologies?
So on the Billy side, I would say, more information is better. Like we've been talking about, the more biological molecules you can see and the deeper you can characterize the biological molecules function in proteomics, metabolomics, lipidomics, glycomics, the more power you have to advance healthcare, right? And so our Billy platform sees more proteins and sees more post-translational modifications. So PTMs are modifications or changes to proteins that occur after proteins have been synthesized or translated in the body. And that can happen to proteins throughout the course of your life. And those changes in the proteins that PTMs or post-translational modifications can dramatically affect the protein's function, stability, and can affect health. And so today's platforms in proteomics do a pretty good job of seeing the proteins, but they don't always go deep enough to see those post-translational modifications or PTMs. And that's what our platform does. It kind of advances and, again, addresses blind spots or breaks through the barriers of today's incumbent instruments. Seeing all the proteins is one thing, but seeing the proteins and the PTMs or the post-translational modifications, that's really what will advance science as a whole, and that's what our platform does, our Billy platform. In terms of our initial product, our MOBI platform, and ways that customers are using it is really in a more targeted way to characterize drugs. As an example, we have one customer or a number of customers in the peptide therapeutic space where you really want to optimize the development of a peptide therapeutic. And a lot of times linear peptide therapeutics can be made into cyclics or circles, because if you take that linear peptide and you put it in a circle, it can have better efficacy, increased half-life, and opens the possibility for oral drug delivery instead of an injectable drug delivery. And so it's not only the amino acid sequence of the peptide, but it's also the structure of the peptide that drug companies spend a lot of time optimizing for better safety and efficacy profiles. And so as an example, when a drug is converted to a cyclic peptide and you break that cyclic peptide up to characterize it, you can sometimes miss site specificity and an understanding of the drug that's critical to its safety and efficacy, and we address those blind spots. So as an example, more accurate characterization of a peptide therapeutic that we do in minutes that takes incumbent instruments, say 8 hours of analysis time per 86 well plate, we can do that analysis of that 86 well plate in minutes instead of a day. And so that just increases, and with more accurate results. So that higher throughput, more accurate screening for large peptide drug libraries, and that leads to better candidate selection and saves money holistically in the drug development process. We have another pharmaceutical, just to give some examples, of pharmaceutical company that was developing a KRAS inhibitor drug, and they knew that there were two forms of this drug that they couldn't separate or identify using conventional techniques. One form is significantly more active and efficacious than the other, but they couldn't separate them in their drug development, and we could. So it's really advancing, developing the most safe and efficacious drugs by being able to better characterize those drug compounds in ways that other instruments can't. There's also on the mechanism of action side. So we have a pharmaceutical customer that's using our MOBI platform to better understand defects in lipid metabolism mechanisms associated with neurodegenerative diseases. So in terms of understanding lipid metabolism, they can more deeply and more accurately characterize the patient biology in this lipid mechanism to better identify the target of the drugs that they're developing to target these neurodegenerative diseases. And so in this case, they were having to use, with the existing platform, it was I think a 2-hour workflow that was missing some of these key lipid identifications. And we converted that to a 2-minute workflow, and we saw more of the lipids that they were trying to characterize to better understand the mechanism of action, and then ultimately develop the right drug target to target that mechanism of action. So they're all, regardless of the drug or the mechanism, they're all variations of the same theme in terms of either seeing an impurity, seeing a form of the drug that they weren't able to see with the incumbent platforms, and then doing that significantly faster from a throughput perspective.
Are there other companies doing similar things to you, or are you kind of out on your own with what you're doing?
Yeah, so there have been, what's been really interesting, coming back to the core technology of ion mobility-based separations. There are companies, and the major mass spec players all have their flavor of ion mobility. And the researcher that I mentioned, Dick Smith, has been critical in the field of advancing ion mobility separations as a whole. Our version, the slim version, is his latest and greatest, and he invented it because he has such a legacy. He's retired now, but had such a legacy in the development of this field as a whole. He knew what the deficiencies of all the predicate approaches were. And it's a really amazing innovator story. He basically took his research team and a whiteboard and said, okay, we know that there are these incumbent ion mobility approaches that he had a hand in inventing most of them and working with other companies to develop most of them. But he also knew what each of their shortcomings were. And so he said, if we developed and just started from a whiteboard and developed a leapfrog ion mobility separations technology that addressed all the shortcomings of what we know from the incumbents, what would it be? And that was SLIM. You know, if you tick through what all the predicates in this field of separation science are, you could check off the deficiencies of each one, and he has taken into consideration those deficiencies and addressing those deficiencies with the SLIM platform technology. Whether it's cost, form factor, complexity, power of separation, you name it, sensitivity, all the lack of resolving power, he pretty much fixed it all with this platform technology, which is really exciting to have exclusive access to harness and productize and commercialize.
As the field moves forward and omics research becomes more central to drug development, how do you see your platforms and your company evolving to meet those needs?
Yeah, well, and that's what's really exciting about it is that, you know, we've been working on productizing, commercializing SLIM for 8 years now. In our earliest days, the industry wasn't ready for it yet. I would say the industry was very largely focused on genomics. And I think now the industry is ready for it, which is an exciting time for us and an opportunity for us. Because if you think about it, genetic mutations maybe are attributed to maybe 20% of disease. 80% is all the other omics. And so for us to have an analyte agnostic platform, meaning we can do proteomics, lipidomics, glycomics, and metabolomics on one platform, and being able to be at the forefront of now the industry is ready and understanding that genomics isn't going to answer all of our questions and we need to have all the pieces of the jigsaw puzzle. We are the leaders in characterizing all those other pieces of the jigsaw puzzle. And so it's a super exciting time for us. And we collaborate with all the mass spec vendors because we don't make the mass spec detectors. We just integrate our technology with their detectors. And so it's a really exciting time for us and them to be advancing the field, and the industry's ready for it, right? The industry now today is ready for it. And I think we're just beginning. So it's super exciting for us.
But what are some of the challenges that you still face though?
Well, look, we're a small company, right? We're a small startup company that works with and is going to market and selling products in an industry against the big players and the large multinational, multi-billion-dollar companies. It's difficult. It is a very capital-intensive industry. Building these instruments is, you know, not inexpensive. It's very capital-intensive. It takes a long time. And it takes a long time to change people's thinking and users' thinking, right? When you said it earlier, when someone's used a separations technology for 35 years, and now you're saying, or longer even, and now you're saying do it in a different way, it just takes a long time to educate people, gain the confidence, make them believers, you know, teach them that there is a different and better way and you can trust it. That takes a long time and it takes a lot of capital. And so it's difficult to survive as a small venture-backed company in a very capital-intensive market that takes a long time to really move the needle and scale. I'd say that's our biggest challenge.
How do you see the company evolving?
Yeah, so I mean, look, our goal is to have our technology integrated with every mass spectrometer, every vendor for every application, because when we integrate it, again, today LC mass spec is here and we integrate our technology in that workflow, we can bring the quality of that analytical result to here across the board. And so that's really what we want to do, is have our technology integrated with all LC mass spec workflows for all vendors, not only in the pharmaceutical industry, food testing, environmental testing, you name it. We can really advance analytical characterization in a way that I think is pretty remarkable and pretty unique.
One thing that's clear about the life sciences is that it's a huge field, and it would be pretty impossible to know everything about every aspect of it. So it's always good to find out about areas you're not quite as familiar with. And so that was a great overview there from Melissa over at MOBILion. So that's it for this week's podcast. If you do want to get in touch with guest opportunities, or if you want to advertise, please feel free to contact us at jim@deeptechdigest.com. We'll be taking a couple of weeks off over the holiday season, not because we want to necessarily, but because the listener numbers always go down. But we're already recording interviews for January podcasts. So if you're thinking about putting someone forward as a guest, please do bear in mind we're already well into January. I should also point out, again, that we do not charge a fee to be on the podcast. We want to provide current, varied, informative guests so people actually listen. What a crazy concept. I should also point out that we will be here again next week for the last one in December on the 19th of December. So until next time, wherever in the world you are, I hope you have a great week ahead. Thanks for listening. And I hope you'll join us again next time for another Base to Base Biotech podcast. The Base to Base Biotech podcast is an Air Coastal Media Limited production.